The second study is from Michael Talkowski at Mass General, and this is the second year of a project, the title of which is CRISPR-mediated molecular dissection of PWS. So we know that there's several genes in the PWS region, as our diagram showed, but we don't know the exact contribution of each of them to the characteristics of PWS. So this team does a lot of CRISPR technology. They're kind of they're a high throughput team that is really technology based, and so they're using CRISPR technology to generate a series of cell lines that have the individual genes in the PWS region cut out very precisely. And the novel thing about this in particular is they're all done on the same background, genetic background, so the same cell, and then they clip out gene 1, gene 2, gene 3. And so the comparison can be very tight. It's a nice comparison about what each gene does individually. So they've had a year of funding, and they have made tremendous progress making large deletions and small deletions of the PWS region. And what they want to do in year 2 is do kind of a gene by gene approach and put it into the same workflow. Their workflow is shown here, where they design the CRISPRs, they make the cells, they characterize the cells, they do advanced genomics, all on the PWS cells. And the nice thing about this is it's building a resource that can be used by all the members of the PWS research community. So they'll deposit that into the stem cell bank that we have, and there'll be very precise excisions of PWS genes. And the Talkowski lab is they're very much involved in other neurodevelopmental disorders, other causes of autism, and other syndromes that occur in humans, and they'll be able to compare PWS to those other syndromes. And sometimes by doing that very large comparison, you can get a real idea of pathways that are similarly affected in PWS and other neurodevelopmental disorders.